Hello, my dear friends. I'm back again with the new topic of endomembrane system. Okay, today we are going to discuss about lysosomes. But before that, let us just recap the things which we have done before. First, let us recap about cell wall. Cell wall basically are basically of three layers: primary, secondary, and tertiary. Tertiary layer is not found in all the plant cells. Okay, it is only found in the tracheids of gymnosperms. What is the plant cell wall made up of? It is made up of basically cellulose, hemicellulose, pectins, and proteins. Okay, now there is a cementing like structure which is named middle lamella. Okay, and after that there is a tubule which traverses through the cell wall. It is known as plasmodesmata, and when endoplasmic reticulum traverses through this plasmodesmata, then it is known as desmotubule. Okay, so in the plant cell, first primary cell wall is made, which is present in the meristematic cell. Okay, and when the cell matures or when it starts maturing, then secondary cell wall is formed, and gradually primary cell wall diminishes. Okay, now there we also studied about the differences between them. Like primary cell wall is thin, secondary is thick. Primary is single layer, secondary is multiple layer. Primary is present at the outermost, secondary is present inside the primary cell wall. Primary cell wall is found in the meristematic cell, permanent cell. Secondary cell wall is not found in the permanent cell. Secondary cell wall is not found in the meristematic cell, while primary and secondary both are found in the permanent cell. Okay, but you cannot say exactly that primary cell wall is absent. It gradually diminishes. Okay, but in mature cell, primary cell wall is absent. Okay, next we discussed about endoplasmic reticulum. Endoplasmic reticulum is basically a system of membranes which provide a network for transportation of materials inside the cell also. Okay, it can also provide membrane to the nucleus at the time of cell division. Okay, it forms a nuclear membrane at the time of cell division. It also helps in making of Golgi apparatus. It also provides raw materials to Golgi apparatus. SER is responsible for lipid and glycogen synthesis. It also helps in detoxification of toxic materials with the help of P450 cytochrome. Okay, RER is responsible for protein synthesis. Endoplasmic reticulum also makes steroidal hormones. After that, we discussed a modification about endoplasmic reticulum. Okay, when a cell is centrifuged, when rotated with high speed, now fragments of endoplasmic reticulum after that we can see, and there are some fragments of ER in which ribosomes are still attached to it. Okay, so those are basically called as microsomes. Microsomes are used by the scientists in the in vitro studies of protein or in vitro protein synthesis studies. Okay. After that, we discussed about the Golgi complex and its function. Okay, Golgi complex was discovered by Camillo Golgi in the year 1898. Golgi complex is also known as the middleman of the cell. It helps in the intracellular transportation of materials, or can say acts like a director of molecular traffic inside the cell. Okay, it is made up of stacks of cisternae, and cytoplasm surrounding the Golgi apparatus is almost clear around it, and it is known as zone of exclusion. You can only see ER around it. Okay, both phases of Golgi complex are different from each other. One is convex, cis or forming phase, and another is concave, trans or maturing phase. Next comes components of Golgi complex. It is made up of cisternae, tubules and vesicles. Now next is the function of Golgi complex. Golgi complex, main function of Golgi complex is basically secretion. Now there are three stages in secretion. First is fusion of raw materials, which is basically proteins and lipids secreted by the RER and SER. Okay, then after the fusion, they are attached with the carbohydrate chains. Okay, which is known as glycosylation or glycosidation. After that, finished products are released from the Golgi complex through the concave phase. Okay, so these are basically three steps in which it is carried out. Now it also helps in the formation of glycoprotein and glycolipid. Okay, it also helps in formation of lysosomes, synthesis of carbohydrates which are used for cell recognition, spermiogenesis and vitellogenesis. Okay, and it also helps in the formation of phragmoplast and cell plate. Okay, at the time of cell division in plant cell, because see, plant cell has a cell wall because of which it is not easy to divide cell. So there are some special formations which are made by the Golgi complex which helps in the cell division inside the plants. After that comes is lysosomes. Lysosomes basically carry 50 different types of enzymes in them, which are after the formation inactive. So it is also known as storage granules. Lysosomes are polymorphic and they are absent in RBCs because there is no digestion carried out in RBCs, whereas it is present in large size and number in the WBCs because they have to digest the antigens or can say pathogens, and they are found rarely in plant cell because plant cell has a large vacuole and digestion is carried inside the large vacuole only. Now you all know that cytoplasm has a pH of 7.2, whereas lysosomes require a pH of 5 in order to activate their enzymes. Their enzymes are basically, I will just name some of them: carbohydrases, lipase, amylase, cellulase, pectinase, proteinase, etc. Okay, so when lysosomes are secreted by the Golgi apparatus, they also have a pH of 7.2, but we have to reduce the pH to 5 in order to activate those enzymes. So pH is basically concentration of hydrogen, okay, concentration of H plus ions. So in order to increase the pH, we use the channel, okay, channel protein, which helps in the transportation of H plus ions. This is also known as H plus protein pump, which reduces the pH from 7.2 to pH of 5 and activates all the enzymes present in lysosomes. See, now transfer of H plus ions is taking place from a gradation of from lower to higher area. Okay, so it is also known as active transport, as we have already discussed about it. So energy is required in it. Now there are basically three types of it: primary lysosome, also known as storage granules; secondary lysosome, or also known as digestive vacuole or heterophagosome; and after that tertiary lysosome or residual body or telolysosome. I will explain you all this one by one. Now see this diagram. Okay, now this is Golgi apparatus transphase. Now see, this lysosome is secreted by it. This is known as primary lysosome or storage granules. Okay, then this is basically phagosome. Food particle is known as phagosome. Okay, then primary lysosome fuses with the phagosome, and then it is known as secondary lysosome or digestive vacuole or heterophagosome. Then digestion takes place inside it and useful products are then absorbed. Okay, then it then forms tertiary lysosome or residual body. There are some waste materials which are secreted by it, then it is known as tertiary or residual body. Okay, then after that this waste material is thrown out of the cell through the process of exocytosis, and all the enzymes are then secreted out, so that is why they are also known as suicidal bag. Okay, now see this another case. This is the primary lysosome secreted. Now suppose there is an inactive mitochondria. Okay, there are many reasons because of which sometimes cell organelles get inactive. Okay, then it fuses with the inactive mitochondria and it digests the inactive mitochondria also. Okay, now this is the part of a cell only, and it is not a foreign particle, so it is known as autophagosome. This is the secondary lysosome. Okay, now this is tertiary lysosome. Useful materials are again reabsorbed and sent for recycling or can say for different purposes, and then through exocytosis useless products are released from the cell. Now see, primary lysosome are the freshly formed rhizosomes. Okay, and they contain 50 different types of acid hydrolases in inactive state. Okay, so they contain these enzymes because an inactive state, so that is why they are also known as storage granules, because they are storing these hydrolases. Second I said is secondary lysosome. Okay, now secondary lysosome is formed when phagosome containing food particle fuses with the primary lysosome. Now this structure is known as secondary lysosome, and when digestive enzymes are activated by maintaining the pH of 5. Now third is tertiary lysosome or residual body. Okay, now food is digested and then reabsorbed by the cell. Okay, then the leftover residues are present. Such structure is known as tertiary lysosome or also can say telolysosome. Fourth is autophagosome, as I have already discussed. This is formed when primary lysosome fuses with any inactive cell organelle. After that comes functions of lysosome. Okay, now basically there are two types of digestion, intracellular and extracellular, and also there is autolysis. In intracellular digestion, there are again two types, heterophagy and autophagy. Heterophagy is basically when some foreign particle enters the cell and it is digested by the lysosome. Okay. So, when foreign particle is digested by the lysosome, it is known as heterophagy, and suppose lysosome is digesting any cell organelle present inside the cell, okay, then it is known as autophagy. In both the cases, digestion takes place inside the cell only. Now, second is extracellular digestion. It means that when lysosomes are sent out of the cell for the digestion purpose. For example, in fungi, lysosomes are sent outside the cell, and after that food particle is digested. And one more example is osteogenesis. See, newborn babies have small bones. Okay, so as we grow, bones also size of bones also increases. So this is basically depositions around the bone. Okay, now sometimes there is more deposition in can say sides or upside. Okay, then osteoclast, also known as bone eating cell, comes here and gives shaping to the bone by eating those extracellular material. Okay. They also secrete the lysosomes outside the cell and then digest these extracellular material. Okay. Now, third is autolysis. Autolysis happens when a cell activates all its lysosomes and releases it inside the cell. Okay. Then that cell is digested completely by the lysosomes. This phenomena is known as autolysis. Okay. Basically, when all lysosomes inside the cell are activated and which digest the cell, it is known as autolysis. Like this is the tadpole, okay, and this is the small baby of it. Okay, so see, it is having a tail and tadpole is not having a tail. So with the help of cathepsin protein, okay, which it dissolves the tail of the tadpole. That is why when it grows through metamorphosis, you do not see the tail here. Okay. Now, one more thing, that how are lysosome, when lysosome starts forming, it basically starts from the endoplasmic reticulum. Okay, because see, endoplasmic reticulum send the materials to the Golgi apparatus, and then Golgi apparatus forms the lysosome. So basically it starts from the Golgi apparatus. Okay, sorry, endoplasmic reticulum. So that's all for today. Thank you.